Hi guys, here's another video which regarding this topic, structure and the function of the antibody. So antibody is a large protein produced by the plasma cells and constitute gamma globulin, and these are called as the immunoglobulin, and it is used by the immune system to identify and neutralize the pathogens such as the bacteria and the viruses. The basic structure of the antibody is here, in which the four type of polypeptides are involved: two antigen and two antibody. The two light chain and the two heavy chain are present here. The Fc represent the fraction of crystallization, while the Fab is represented by the fraction of antigen binding site at the part antigen will bind with antibody. So structure is the antibody are heavy globular plasma proteins or the glycoproteins, with each is heterodimer with a molecular weight of the 150 kilodalton. An antibody is composed of two identical heavy polypeptide chains and two light chain bonded by the disulfide linkage. So heavy chain. 5 types of heavy chains are present. Each heavy chain has two regions, one constant and the other is variable. 5 types of heavy chain are as follows, like alpha, delta, epsilon, gamma and the mu. Light chain, all the antibody have one of the two kinds of the light chain and has two successive domains, that is constant and variable region H. The length is about 211 to 217 amino acids. The two types of light chain is kappa and the lambda. Immunoglobulin G, that is, it is most abundant and smallest size. They make up approx 80% of the serum antibody and a half-life of the 7 to 23 days. IgG3 binds to the Fc receptor of the phagocytosis, immunity to newborns, and the neutralization of the toxin. So figure of the IgG. Then IgA makes up approx 6% of the serum antibody and the half-life is about 5 days. It is second most abundant antibody and may called as the copro antibody, as may appear in stool. Function of the IgA is it is a secretory antibody effective against virus that causing influenza, production to infant gut. IgD makes up approx 0.2% of the serum antibody, and it function are the B cell activation, act as a receptor for antigen binding. IgE was discovered by the K. Isijaga in 1966. It is very low concentration in blood. It binds to the Fc receptor on basophils and mast cells, the least amount of the antibody, and highly sensitive to heat, associated with the allergy and the fight against parasitic infection. IgM makes up an approx 30% of the serum antibody. It is largest size antibody. IgM is the first antibody who synthesized within developing fetus. So it may be asked in the examination. So function is activation of the classical pathway, defense against multivalent antigen, act as opsonine. The light chain, the kappa and the lambda chain family contains V, J and C gene segment, which after rearrangement encode variable region of the light chain. We have already seen in detail in the previous videos of the light chain and the heavy chain. So this is about the structure and function of the antibody. Thank you and keep watching.